welcome to today's episode of our Discover Molecular Interactions webinar series. My name is Vivian. I am responsible for scientific communication at Bruker Biosensors, and I will be your host today. And in this webinar series, we explore different molecular interactions each month, and we discuss assays to analyze them. And in previous episodes, we've already talked about how to use switchSENSE to characterize ternary binding, for example, for PROTAC molecules. And we often get asked how these biophysical characterizations also relate to cellular degradation. So I'm very happy to introduce today's topic, which is an exciting collaboration project we did together with Novalix. And we will show you a complete workflow starting from a DNA-encoded library screening to identify binders to ternary complex analysis all the way to cellular degradation. And I'm happy to introduce today's guest speaker from Novalix, Renaud Morales. Hi, Renaud.
Hi, everyone. Hello, everyone.
Renaud did his PhD in protein crystallography at the IBS in Grenoble, and then he moved to England for a postdoc at the Cancer Research Institute. And then after working at Pfizer as well as at Sanofi, where he also developed the biophysics platform, he now joined Novalix. And there he's currently leading a team for structural biophysics, including mass spectrometry, NMR, and also SPR. So now I would like to start by giving you a brief introduction into the biosensor methods that will be shown in this project so that we are all up to speed and we all know the basic measurement principle. If you have any questions during the talk, please feel free to use the Q&A function, or if that is not available in your app, you can also use the normal chat function. And alternatively, you can also talk to us directly after the presentation in our Q&A session. Just raise your virtual hand and I can unmute you and you can pose your questions directly. Now, if we look at the drug discovery process, the biophysical technologies can be used in many different steps to advance the development of new drugs. That can be from high-throughput screenings in the hit generation steps to the more in-depth characterizations in the hit-to-lead and lead optimization steps, all the way to in vitro cellular assays, for example, with interaction cytometry, to characterize binding directly on cells. And we are going to discuss mostly the SPR steps and switchSENSE for more in-depth characterization. So if we go to the next slide, we will see our SPR64 Triceratops instrument, which is the SPR instrument that was used in these studies. And the Triceratops SPR64 is our high-throughput SPR device. It has many unique design features. I will not get into all the details shown here, but important points are, for example, its high sensitivity imaging system and also the compatibility with different plate handling robots. And what is very important, and I would like to point this out today, is the microfluidics and the rotatable manifold. So first of all, it's very low maintenance. You don't need to exchange any IFCs, but also the manifold is very special. And on the next slide, you can see a close-up of the manifold, and it's rotatable. And this will allow you to get unmatched throughput in the SPR range. So you can immobilize up to 8 different targets in the vertical direction and analyze up to 8 different analytes in horizontal direction. And this will give you 64 kinetic characterizations simultaneously. So this is very unique and this will be used in the following examples. Now, the second technology I would like to briefly introduce to you is switchSENSE. switchSENSE is based on a chip that has DNA-based nanolevers on its surface. And these DNA-based nanolevers are equipped with a fluorescent dye, as well as your protein of interest. And now on the next slide, you can see how a typical measurement would look like. So upon the injection of analyte molecules, you see a change in fluorescence due to quenching effects on the fluorescent dye. And when we flush and buffer instead and the analyte dissociates again, you will see the fluorescence being restored to its baseline value. And from these changes in fluorescence, we can then derive on rates and off rates. And since the whole system is DNA-based, also the regeneration of the chip surface for further measurements is very straightforward. So this is like the basic principle, how these systems work. and the specific use cases and how you can use them for an in-depth PROTAC characterization will now be shown by Renaud. So now I hand over to you, and I'm looking forward to your talk.
Thank you, Evan. So first of all, I would like to say I'm very happy to be here today and to present this webinar. Even if, to be honest, I would have preferred to see Anna Bonneau presenting the slide she prepared herself. But unfortunately, Anna not feeling well today, so I replace her, and I will do my best to fill in for her and presenting this great collaboration we have with Bruker. Please bear with me. As you understand from the title, we will talk today about PROTAC. So targeting protein degradation, as you probably know, has rapidly emerged as a powerful new therapeutic approach, with PROTAC being the most widely used and advanced strategy. The project I will present today is an internal project we have developed at Novalix, where we use, in fact, BRD4 as a target, the protein of interest. And we go from the DEL screening to find a hit, transform this hit into a PROTAC, and characterize the PROTAC molecule in terms of biophysical characterization, meaning binary, ternary, complex formation, and also in cellular degradation. So for the sake of time, I will not develop the early stage of this project, meaning the design of the library for the DEL, the hit triage for the DEL screening, but I will only focus on the last part, which concerns what has been described by Vivian, the use of the Bruker biosensor techniques. Okay, so I will show you. Okay, so DNA-encoded library screening has emerged also as a cornerstone of technology in drug discovery, enabling the exploration of extensive chemical space through libraries of million, even billion of compounds with optimized PCAM properties and diverse chemotype. This approach surpasses the classical HTS in terms of throughput, so it's a very interesting approach. So in this project, we decided to use the DNA-encoded library screening because also the DEL-derived hits represent particularly valuable starting point for PROTAC development. Indeed, we can use an exit vector, I mean the DNA tag attachment, and we can use this to guide the conjugation of the I3 ligase moiety. So it makes sense to start from a DL screening for PROTAC optimization. So the goal of this project was to find specific ligand for the BRD4 D1 domain. And so during the screening on the DEL, we utilize a construct of BRD4 where the BRD2 domain has been mutated. And this mutation interferes with the binding of a reference compound in the active site of BD2 domain. For this internal project, we screen a subset of the Novalix library, Novalix or DEL library, which contain the subset 30 million compounds. The analysis of the hit on during the DEL has been performed internally, and around 500 compounds were selected based on the enrichment and copy number. Based on that, we decided to synthesize of DNA molecule. Only 8 were synthesized. And they present some attractive PCAM properties, as you can see in this table, in terms of molecular weight, hydrogen bond donor acceptor, or P or topological surface area. So nice properties, but the idea was also to test these 8 different hits synthesized of DNA in parallel in a SPR assay to determine the affinity of this compound, and also in a threat binding affinity assay. This is shown in this table. In this table, you can compare the data we observe, for example, in the BRD4-D1 in SPR with the IC50 obtained in this threat binding assay. And there is a quite good correlation. Just to give you an example, with this hit number 1 here, we obtain this kind of sensogram with a very square shape. And if we compute the affinity KD at the equilibrium, we obtain a KD around 1.4 micromolar. What was also very interesting with this hit is the selectivity we observed. As you can see, for BRD4, sorry, D2 domain, we obtain a KD which is around 90 micromolar, and so we observe a very nice selectivity already. It was expected because I mentioned earlier during the screening, the DEL screening, we use a construct where we should obtain only specific binder for the D1 domain, but it's good to confirm that. Then the idea was to use this hit1 I described and to transform this molecule into a PROTAC degrader without further optimization. That means we will create PROTAC molecule using this hit number 1 here, add a linker, and another moiety. In our case, it was a Cereblon moiety. So we use a toolbox we have at Novalix, where we can have a different length or different type of linker, and we attach on the other part the high-free ligase you want. So only 5 molecule PROTAC molecule were synthesized using this approach. So the idea here for this project was, okay, are we able, starting from a low micromolar range hit from the DEL, are we able to transform this hit into a PROTAC molecule where we are able to observe the degradation in cell? That was the goal of this project. So as mentioned, for the binary interaction, we use the Triceratops systems. Due to this high throughput, we were able to test not only the BRD4 domain, but also different bromo domain and different isoforms. Just to give you an idea, for 4 protein immobilized and 8 molecules can be tested in parallel. This represents around 28 hours runtime. So let's say in 24 hours, you have your full information about the selectivity of your compound. So it's a very impressive machine. Here on this view, you have the overview of all the sensorgram we obtain. So it's pretty small, but I will describe a little bit more in detail later on. You have the 5 PROTAC molecule here, tested on the different bromo domain, BRD2, sorry, BRD3 and BRD4, and for each of them we have the D1 domain only, D2 domain, D2, D1, and for BRD4 the only difference we have BRD1, BRD2, and also domain 1 and domain 2, sorry. In addition also to that, we immobilize the Cereblon protein on the chip as well. We also tested some reference compounds, some reference PROTAC molecules, which are known to interact with the BRD4, like, for example, the NZ1, where we observe a clear binding on BRD4. But this PROTAC molecule do not interact with the Cereblon because it has a VHL moiety. This is why we do not observe any interaction with the Cereblon. Another way to look at the, this interaction is to put the data on a heat map where the affinity, the KD affinity, is indicated with a color code. So the blue is the most potent inhibitors, and the red are the less potent. And it's very interesting to use this approach or this heat map to compare the profile. So let's go a little bit into more detail. In terms of the PROTAC, what we can see is that all the PROTAC tested are selective towards the BRD4 D1 domain. It was expected, but again, it's good to see it. Compare, for example, to the BRD2. If you look at just this heat map, you can see that all the PROTAC are more active on the D1 compared to the D2. You can see it also on the sensogram, actually. For example, if you compare now with the BRD4 D1, D2 construct, you can see that we have similar profile. That means that the BRD1 drives the interaction of this PROTAC molecule. Another interesting compound is the PROTAC4. PROTAC4, in fact, is the highest has the highest affinity to BRD4. You can see with nanomolar, you can see nanomolar affinity for D1 and D1, D2 domain, and around 10 nanomolar, sorry. But what is also interesting, this compound has a lower affinity to the Cereblon moiety compared to all the other PROTAC molecules. I have to mention here that this molecule, this PROTAC4, is the PROTAC which has a shorter linker. So it's maybe this shorter linker do not allow some flexibility, might be important for the interaction with Cereblon and interfere with the binding of Cereblon. That's one hypothesis. If we look at the cross-family selectivity, we can see that all of the 5 PROTAC are comparably unselective towards the BRD2 D1 domain. Yeah, they almost interact with all. PROTAC 1 and 3 are the most selective, while PROTAC 2 interact with almost all the BRD domain. So if we summarize what we have observed with the SPR characterization for the binary interaction, we have shown that, and we have confirmed what we are expecting from the DEL screen, that all the compound showing some selectivity towards the BRD4 D1 domain, and the PROTAC 1 has the highest affinity to BRD4, but show more than 40-fold lower affinity to Cereblon compared to the other. PROTAC 4, we will discuss this molecule later on because it seems to be very different compared to the other. So it's good to have this information on your PROTAC molecule, how they bind, how strongly they interact with your protein of interest or with the ligand, but it's even better to have the information of the formation of the ternary complex. And this is where we use the switchSENSE. And in this case, we use the Y-shaped system of this switchSENSE, where you can, in fact, functionalize on the green arm your protein of interest. And in the red arm, you will attach or immobilize, functionalize your Cereblon moiety. When you add your ligand, your PROTAC, you will expect the formation of the ternary complex. And as the arm are very flexible, you will put in proximity the two protein in a fret condition where you will observe a fret signal. So you will observe this formation of the ternary complex using this system. So it's a very nice and well-designed approach for this. So we apply it again on the different PROTAC molecule. We have functionalized with different domain of BRD4, so D1, D2, and D1, D2. So on this part, you observe the formation of or not of the ternary complex, and you still can see when you look at the green signal, you can still confirm the binary complex in your molecule, and here the BRD41. If we look into detail, what we can conclude when you look at this sensogram is that we observe for PROTAC1, PROTAC2, and PROTAC3 very strong ternary complex, which are mediated through the interaction with the D1 domain, because you can see that when we functionalize with only the D2 domain, we are not able to detect any ternary complex formation, which is not the case with D1 or D1-2. We observe a clear, a very fast association and a quite long dissociation for this molecule. PROTAC5 is pretty similar, say, even if we detect faster count and faster count for this molecule. Interesting compound again is PROTAC4. In this case, we observe only a very weak ternary complex formation. As you can see, the curve also is quicker, but we will come back to that. And one thing which was very interesting is for this compound, we observe an anti-quenching signal when we look at the green signal, but the only PROTAC molecule will show this behavior. All the other, you have this negative signal, and here we have this positive signal. So probably linked to a different binding mode or different conformation of this PROTAC molecule. We can also try to look at the data differently. We can try to put the count and curve of each PROTAC molecule on this iso affinity plot. And what you can see is that, actually, I should mention that the circle represents the data we obtain on BRD4 D1 domain, and the square like that are the data we obtained for the BRD4 D1 D2 domain. But what we can see is that for PROTAC1, 2, and 3, they all cluster together with very similar count curves. And the PROTAC5, which is this one in green here, we observe, as I mentioned earlier, same behavior, but a faster count and faster count. What is very interesting here is to see that this PROTAC4 do not belong to the same cluster. It shows, in fact, a very curve which is 10 times faster compared to the other. And as you will see in the next slide, we do not observe cell degradation for this compound compared to the where we observe the degradation. So this PROTAC4, as I mentioned here, is a shorter linker for this molecule, clearly has an impact on the ternary complex formation. This is described also with this anti-quenching signal we observed. And to confirm that, or to look a bit more in detail, we also perform some conformational analysis using the switchSENSE. This is something we can also apply. And you can see that the signal we obtain for PROTAC4 here is very different to the signal we obtain for PROTAC3, for example, where we have a very strong compaction of the structure upon the PROTAC4 binding. So again, a behavior for this PROTAC4 which is completely different. Change the slide. Okay. So an essential step toward advancing PROTAC development is, of course, the implementation of a robust and a quantitative degradation assay in cells. So this is why we develop internally this assay. It is a cellular degradation assay where we quantify the endogenous BRD4 level in LR cells. Okay, and we tested at different time point at 6 hours, after 6 hours, and after 24 hours treatment. What you can see here is a percentage of BRD4 available in the cell when we treat the cell with different PROTAC molecule. And we had also the MZ1, for example, response PROTAC. And you can see that even after 6 hours, we start to see a degradation for some of our PROTAC molecule, which it was very interesting result for us. And in fact, the PROTAC3 already show 98% percentage degradation of BRD4 after 6 hours. So it was very nice. If we compare with EDZIM1, which is almost 99%. Okay, at 10 macromolar, we are at 33, but it's already quite good. Because let me remind you that we start from a molecule, a hit from the DEL in a low micromolar affinity, and we do not try to improve the affinity of this moiety for the BRD4. So we start from a very crude, I would call it like that, crude hit without further optimization, but we are still able to detect the degradation in cells. So we were very happy about this result. What is interesting also to point out here is PROTAC4, as discussed already. PROTAC4 is in red here after 24 hours, and that's the only compound or PROTAC molecule where we were not able to detect any degradation in cell. And is probably related to the fact that for this compound, we detect only a very tiny ternary complex formation. What we did also, in addition to this experiment, and in order to understand the mode of action of PROTAC3, we in fact performed some competition, the same assay, but in presence of inhibitor known to inhibit the methylation or the proteasome or the Cereblon. So we tested the PROTAC3 in presence of each of these reference compound. And as you can see, the BRD4 degradation is completely reduced. So this clearly indicates that the PROTAC we were able to create or induce the degradation of BRD4 using Cereblon recruitment and the ubiquitin proteasome pathway. So it was very important for us to confirm. So this is the last slide. Just I hope I've been able to show you the workload we have applied, starting from a DEL screening up to the confirmation of cellular degradation. I've shown that using the binary, the Triceratops system, we were able to confirm the selectivity among the different PROTACs for the different BRD moiety we tested. And using the switchSENSE, we clearly investigate the ternary complex formation. It's a very nice system for that. And the cellular assay we have put in place allows us to confirm the Cereblon recruitment and the application of the ubiquitin proteasome system. So a complete internal project that can be applied for other projects, of course. And then just before finishing, I just want to acknowledge a lot of people from Novalix, but also from Bruegel. We have the SPR team, but we have also the MS team. I was not able to describe the work we did in MS, but we performed some native MS showing formation, also the ternary complexes in TMS. The chemistry team for the off DNA synthesis, of course, the protein production, we need protein, pure protein, so it's very important. Of course, the DEL screening, and also the analyze of the heat and so on. And also, of course, the pharmacology, I mentioned this work. And of course, Cyril and Alice for their work on the system I described today. And thank you for your time, and happy to answer your question.
Thank you very much for this great comprehensive work you showed us today. Yeah, are there any questions? Please feel free to use the chat or the Q&A feed, or also raise your hand and talk to us. So maybe let me start with the first question. Did you see any avidity effects also when you compared like binary and ternary interactions of the PROTAC molecules?
Yes, of course. This is something we have to check. Let me be moving back a slide because I'm probably clearer on this view, where you see the avidity effect. In fact, where we compare, for example, for PROTAC 1, you have the count and calf for the DB1 domain. And when you have the Cereblon, you can see that we observe a strong, for example, in this case, a strong diminution of the curve, and so the KD is much better. I think the KD is right in here. We go from 500 nanomolar up to 60 picomolar. So a huge impact of the proximity of the Cereblon. This is something we observe.
Mm-hmm. Thanks. And do you know if like the ternary complex is important in many upcoming and promising therapeutic modalities? So there's not only PROTACs, there's all sorts of targeted degradation methods right now, like glues. And can you envision to like how these biophysical approaches can also be helpful in other modalities and aspects of this?
Yeah, you're right. PROTAC is only one part of it. I think for molecular glue, definitely this is something that the switch can be very useful to tackle this kind of approach as well. Pretty sure you have already tested that project, but yeah, for sure. Yeah. I don't know, for the SPR, I don't know, make more sense to use the switches for molecular glue.
Mhm. Yeah, I think this also nicely shows how different biophysical technologies have to be combined to really have like the full picture, and how you can use different advantages of different systems at different steps of these development processes instead of just relying on one system, right? I mean, that's
Yeah,
always important
absolutely.
nowadays. Yeah.
We always try to perform, in parallel different approach. So the big picture, I would say, is always a good idea. Yeah, sometimes if you stick with for example, with PROTAC 4, you stay with your binary information. We say, okay, that's the best compound we have. Let's go with this one. And in fact, you will have no no degradation. So it's always good to have all the information.
Mm-hmm. Yeah, that's true. Also, cell experiments are probably pretty difficult and more expensive, so it's nice to get some more information in advance, right? To know which candidates you want to take into like further steps that might be more time
Yeah.
consuming.
Something for the threat assay, maybe you need to, I don't know if the kit is available. Maybe so here you are free. Just need to mobilize your protein.
There's a question about how you confirm the formation of the ternary complex. Could you explain this again, please?
Okay.
Mm-hmm.
I think we use this one. Yeah. I think the switchSENSE is very nice in this way. This is where you are able to, if you detect this threat signal, this close proximity assay, in fact, is based on the threat signal. The threat is only you can observe this signal only if the two proteins are close enough to promote this threat signal. So if the ternary complex is formed, you observe this signal. So this is how we say, okay, we have the ternary complex. We also confirm it. I can, if I have a few minutes, I can show one more slide. We confirm it also by SecUV native MS internally, where, in fact, for example, you on the sec dimension, you observe the UV, and so here you have two peaks, one for the Cereblon and one for the BRT4. If you inject your PROTAC, you observe the shift of this peak almost disappear, not completely, but shift at least. And this one also decrease, which is not the case, for example, for PROTACs 4, where here you still have the same peak at the same retention time. So for the PROTAC 4, no recruitment between two proteins, but for PROTAC 1, we do observe at least maybe a partial recruitment, but we do observe the formation of a complex. And how we know that is we can, using this SecUV native MS system, you can have the MS signal for each peak. And if you look at the MS signal for this peak here, you have these new peaks here, which correspond to the ternary complex. So this is how we have the full picture.
So you can like additionally confirm ternary complex yes, no by mass spec, and you can get like the kinetics of how fast is the ternary complex formed and how fast is it dissociation with like switchSENSE. So.
Yeah, absolutely. You're right.
Okay. I don't see any further questions. Please also feel free to reach out to us after the webinar in case something comes up. You can reach us at info.biosensors@bruker.com. And we are always happy to also do technology demonstrations. So if you have any PROTAC projects and you would like to see ternary complex formation, you would like to test if the Y structure assay or the Triceratops could be something that works in your workflow, we are always happy to come to you and you can test the system with your own projects. So please reach out to us and also follow us on social media to find out about upcoming episodes of this webinar series or to see any recordings of our previous webinar series. And with that, I thank you all for your attention. Thank you for joining us. Thank you again, Renaud, for joining us today.
Pleasure.
And I wish you all a great rest of your day.
Thank you.
Bye-bye.
Bye-bye.